Hello everyone, it is my great pleasure to introduce our latest work on explicit ion modeling of physicochemical interactions for chromatin organization. This manuscript has just been published on the eLife website. In this work, we introduce an explicit ion model to rigorously characterize the nucleosome interaction and chromatin structure in a nuclear environment. In this model, the chromatin was simulated as a residual resolution coarse-grained model, and the ions are introduced based on their concentration. First, we validate the model by measuring the DNA binding affinity for a series of protein DNA complexes. And we find that the model can rigorously capture this protein DNA binding affinity. And second, we validate the model by looking into this unwinding free energy of a single nucleosome. And we find that the model can not only quantitatively reproduce the unwinding free energies of the outer layer DNA, but also inner layer DNA. Additionally, we find a series of contact ions would bind to the inner layer DNA as they start to unwrap. And that means that there are additional entropy contribute to this inner layer DNA wrapping as these contact ions start to be released. With this validation, it gave us the confidence to further look into the nucleosome-nucleosome interaction. Before our simulation, there are a couple of experiments probing the nucleosome interactions. Especially a magnetic tweezer experiment found that this nucleosome interaction features 14 kTs interaction strength. However, a more recent DNA origami-based experiment found this nucleosome interaction is much weaker. To reconcile such difference, we use our simulation to probe these nucleosome interaction at different ionic concentration. One thing to note is that this DNA origami experiment features a somewhat restricted motions of nucleosome. And to simulate such a scenario, we impose a series of restraints in our simulation where the nucleosome can only lay on top of each other, with one of them moving only on an arc that is a certain distance away from the origin. And we find such a restraint can explain the difference in different experiments. When we have a restraint, the system features about 2 kTs difference moving away 2 nucleosomes. But removing such a restraint can recover 14 kTs of interaction strength. Having been able to reconcile such a difference, we continue to use this model to examine the interaction strengths between 2 nucleosomes in a physiological nuclear environment. And we find there are about 9 kTs difference for nucleosome interactions. And these intrinsic interactions would be very important for chromatin organization. Also, we find an interesting orientation dependency. When 2 nucleosomes are very close to each other, they feature a face-to-face contact with 2 nucleosomes laid on top of each other. When they are slightly farther away, they face a perpendicular motion with one nucleosome laid perpendicular on top of the other one. Only when they are moving very far away, these 2 nucleosomes interaction lose orientation dependency. And one advantage of this residual resolution model is that we can utilize it to study different impact of chromatin factors. In reality, nucleosomes are connected by extra DNA known as linker DNA. To study that, we added 10 base pairs of DNA on each side of the nucleosome. And we find this additional DNA would weaken this nucleosomal interaction by adding repulsive interactions. But this interaction can be further compensated by the addition of linker histone. Linker histone is a very important chromatin factor modulating chromatin structures, especially for higher eukaryotes. And we find the addition of linker histone would strengthen these nucleosome interactions. Especially the disordered tails of the linker histone would interact with this nucleosomal DNA, and by doing that, it will strengthen these nucleosome interactions. And finally, we move onward to study the ion impact on chromatin organization. Interestingly, earlier experiments find that a similar concentrations of monovalent and divalent ions have different effect on chromatin. With 5 millimolars of sodium, it can feature a very expanded chromatin structure, but with 1 millimolar of magnesium, the chromatin features a very compact state. When we simulate the system with our explicit ion model, we find indeed the model can reproduce this different effect. Especially at 1 millimolar of magnesium, the chromatin features a very compact fibril-like state. A further investigation finds that this magnesium significantly concentrate on top of these nucleosomes. And by doing that, this bound magnesium would neutralize the excess charge to compact the chromatin. As a comparison, sodium ions would only function as a cloud to screen this DNA repulsion. As a summary, we utilize an explicit ion model to probe the physicochemical interactions of a chromatin in an in vivo environment. And we find a strong orientation-dependent interaction intrinsic between nucleosomes. And the addition of this linker DNA would weaken such an interaction, but it will be further compensated by the addition of linker histone. And finally, we find that this magnesium is more effective in terms of condensing the chromatin structure. For more details of our model and its application, please check our newest manuscript published on the eLife website.